Hey guys, today we're going to be talking about using CRISPR gene editing techniques to change pigmentation in flowers. Specifically, we're going to be talking about Japanese morning glories, which are usually violet. I know it's pink in the video. And we're going to change them from violet to having a lack of pigment. We're going to be doing this by using CRISPR, which is a super cool technique. Specifically, we're going to be talking about the CRISPR and CRISPR-associated protein 9 technique. CRISPR can be used to create targeted mutagenesis in a DNA sequence to remove a segment of DNA and render the enzyme that DNA creates inactive. And that's what we're going to be talking about doing to reduce pigment in flowers. In Japanese morning glories, the DFRB DNA sequence codes for an enzyme that makes anthocyanin, which is a water-soluble pigment that makes pink, red, purple, violet colors in the Japanese morning glory. And when we make the DFRB non-functional, it won't make anthocyanin, which will make no pigment in the flowers. See, it's like kind of sad because we like pretty flowers. And how are we going to do this? Through CRISPR-Cas9 gene editing. The CRISPR gene editing technique uses a guide RNA, or sgRNA, which consists of two parts, the crRNA, or other known as the CRISPR RNA, which is a sequence of RNA that is complementary to a 20 nucleotide region in the target sequence, which will be in the DFRB DNA sequence. The other part is tracer RNA, or transactivating RNA, which forms a series of hairpin loops which function as a binding spot for the Cas9 enzyme so they can form a complex together before they bind the DNA. The 20 nucleotide region that we're going to be targeting on the DFRB DNA sequence is on the 4th exon, and it codes for a catalytic region of the DFRB enzyme. We're going to use this region because when it's removed, it'll render the DFRB enzyme non-functional, which, if you remember, a non-functional DFRB enzyme makes no anthocyanin, which makes no pigmentation. A problem that arises is that the sgRNA can be complementary to different regions that aren't on the DFRB sequence, or at least won't have the same function when removed. And how we get by this is that there's a specific sequence that follows it called the PAM sequence. The PAM is recognized by Cas9, which has two lobes: the recognition lobe, which consists of Rec1 and Rec2 and binds to the tracer RNA, and the nuke lobe, which consists of some endonucleases and the PAM interacting domain, which will recognize the PAM that is specific to the target sequence that we're trying to cleave. Between the complementarity in the sgRNA and in the PAM interacting domain, we can recognize a sequence with high specificity and thus cleave it. The cleavage process occurs using the nuke lobe, which consists of two endonucleases, RUVC, which cleaves the complementary strand to the sgRNA, and HNH, which cleaves the non-complementary strand. And between the two of them, they make two double-stranded breaks, which remove that sequence completely. Once the target sequence has been removed, the cell's repair mechanisms will come in and repair the DNA using mechanisms like non-homologous end joining. The removal of the target sequence makes a DFRB enzyme that makes no anthocyanin, which results in no pigmentation and a sad, sad, colorless flower. Now let's review what we've gone over thus far. First, DFRB makes an enzyme that makes anthocyanin, and that results in pigment in the Japanese morning glory. We went over how to use CRISPR and Cas9 technology to remove a segment of DNA from the DFRB sequence and make that enzyme non-functional. The result of this is that we take a Japanese morning glory that would be normally violet and we make it pigmentless or white in the stem and the leaves and the petals, which is sad but super cool. Now you know how to edit a gene in an ornamental flower. Thanks so much for watching, everybody, and I hope you all have a great day.